After studying this module, you shall be able to know enzyme catalyzed reactions are similar to chemical reactions wherein the rate of reaction is proportional to the concentration of reacting substrates. The number of reacting substrates decides the order of the reaction. Enzymes bind to substrates showing saturation kinetics. The rate of an enzyme catalyzed reaction or its activity can be measured by a variety of sensitive assays. All living organisms use enzymes to catalyze the numerous reactions in their cells. One of the important features of enzyme catalyzed reactions is the rate of the reaction or activity of the enzyme. This activity is useful in deducing what the role of the enzyme is in the context of the biological pathway or metabolic pathway. For example, is that particular reaction being regulated either by increasing or decreasing the activity of the enzyme, and why? Under certain stress conditions like exposure to xenobiotics, which means drugs, antibiotics, an enzyme in the liver related to detoxification, namely cytochrome P450 monooxygenase, increases in activity. An assay of this enzyme could indicate that the organism has been exposed to xenobiotics. There are several such situations an organism is exposed to which necessitate an enzyme assay. Hence, an analysis of enzyme assays would be pertinent. What are enzyme assays and how do we measure substrates and products? The aim of an assay for enzyme activity is to determine the rate at which product is formed or the rate at which substrate has disappeared. Consider a reaction of substrate S forming a product P. If the reaction rate is proportional to the concentration of substrate S, or V is equal to K into bracketed S, wherein V is the velocity of the reaction, K is the first order rate constant. For a reaction S1 plus S2 giving rise to P1 and P2, S1 and S2 being substrates and P1 and P2 being products, this reaction being a second order reaction, the velocity V is equal to K prime into S1, S2, both bracketed because it's the molar concentration. Therefore, the reaction is a second order reaction and K prime is the second order rate constant. Many enzyme catalyzed reactions generally fall into first or second order rate reactions. The velocity V is measured as the disappearance of substrate S with time or the formation of product P in a given time, or V is equal to bracketed P or V is equal to minus substrate, the negative sign before S indicating the disappearance of substrate. In most cases, as the concentration of substrate is much higher, it is easier to determine the appearance of product, although as the stoichiometry is 1:1, either product appearance or substrate reduction is equivalent. In an enzyme catalyzed reaction, the velocity of the reaction is dependent on the substrate concentration. Consider the figure plotting the velocity V on y-axis versus the molar concentration of S substrate on the x-axis. This graph was first determined by Michaelis and Menton in the 1900s. It is apparent from the at lower concentrations of substrate, the velocity is proportional to substrate concentration, and at higher substrate concentrations, the graph becomes parallel to the x-axis, indicating saturation kinetics or Vmax. Hence, while measuring enzyme activity, it would be best to assay at Vmax using high substrate concentration to avoid variation in velocity due to changing substrate concentration. It would be pertinent to point out that as an enzyme acts on a substrate, the concentration of the latter would decrease. Therefore, all assays for substrate or product should be made before 10% of substrate is consumed to avoid an underestimate of enzyme activity. The velocity on the y-axis of the should therefore be replaced by V0, indicating initial velocity, during which period at least 90% of substrate is intact. Now let us consider what are the enzyme activity units. Enzyme activity is expressed in two types of units: international units or IU and ketals. An international unit is the activity of an enzyme which converts 1 micromole of substrate to product in 1 minute at 25 degrees centigrade and normal atmospheric pressure. A ketal of enzyme activity is the conversion of 1 mole of substrate per second under the same conditions of 25 degrees centigrade room temperature and 1 atmospheric pressure. In most measurements of enzyme activity, it is more convenient to use IU, namely international units, as the concentrations of metabolites in cells are in the micromolar range. Let us now consider the various types of enzyme assays. 3 major types of enzymes assays are there, depending on the substrate or product concentration range and the ease with which the change in substrate or product concentration can be measured. These can be classified as follows: fixed time assays, continuous and coupled assays. Fixed time assays. In this type of assay, the enzyme and substrate are mixed, and after a fixed time, the assay is terminated by changing the conditions such as adding base or acid to terminate enzyme activity, and the product formed is measured by a sensitive method like spectrophotometry, fluorimetry or radioactivity. The method selected to measure the product or in some cases the substrate would be based preferably in measuring either one in the presence of the other. For example, in the assay of the enzyme acid phosphatase, a colorless substrate para-nitrophenol phosphate is used, which when acted upon by the enzyme gives rise to a colored product, para-nitrophenol. Hence, the amount of colored product can easily be measured in the presence of the colorless substrate using a spectrophotometer. In the figure, you can easily see how this could be done. What is a continuous assay? In continuous assays, instead of terminating an enzyme reaction after a fixed time, the formation of product or disappearance of substrate is continuously monitored. Again, this is possible if the measurement of substrate or product concentration is possible in the presence of the other. An example of this type of assay is the oxidation of alcohol to acetaldehyde in the presence of the enzyme alcohol dehydrogenase and coenzyme nicotinamide adenine dinucleotide oxidized or NAD+. As indicated, since the stoichiometry of the reaction is 1:1:1 between alcohol, NAD and acetaldehyde, the conversion of NAD+ oxidized to NADH reduced can be followed continuously by placing the assay mixture in a cuvette, which is a holding glass body, which is then placed inside a spectrophotometer and monitoring the increase in absorbance at 340 nanometers. Observe the graph. NAD+ has no absorbance at 340 nanometers unlike NADH, and therefore if the assay is monitored at 340 nanometers, then the amount of NADH formed, which is stoichiometry equivalent to the amount of NAD+ reduced, which is then equivalent to alcohol, the substrate, can easily be monitored. A third type of assay is a coupled assay. In cases where the enzyme reaction has an unstable product, the assay would be difficult to monitor under usual laboratory conditions. In such cases, the first reaction is coupled to a second faster reaction using another enzyme, and the second product, which is stoichiometrically equivalent to the first, is estimated. The scheme for such a coupled assay would be as A, which gives rise to B by the first enzyme 1, and B giving rise to the product C by the second enzyme 2. The measurement of the unstable product B could be avoided by measuring the more stable product C as long as the stoichiometry of B and C is equivalent. The regularly used assay for serum glucose or cholesterol is based on the above scheme. Note, both these assays are very important in the pathological lab which routinely measures serum glucose as a sign or diagnosis of diabetes or cholesterol, which has become important in the diagnosis of heart disease. In either case, that is serum glucose or serum cholesterol, the substrates are oxidized, releasing hydrogen peroxide, which is unstable and therefore is made to reduce a colorless substrate into a colored product by a second reaction which uses peroxidase, which acts on hydrogen peroxide releasing oxygen. The released oxygen oxidizes a dye whose abbreviation TMBZ stands for tetramethylbenzidine diamine, which turns blue in its oxidized form and can be measured spectrophotometrically. Note that the reactions glucose, which is oxidized and can be measured spectrophotometrically. For example, in the oxidation of glucose, reaction 1 is the oxidation of glucose to gluconic acid and hydrogen peroxide in the presence of the enzyme glucose oxidase. Reaction 2, which is coupled to the first reaction, is the breakdown of hydrogen peroxide by the enzyme peroxidase, which then oxidizes TMBZ into a dimer, which then has a blue color. So, the monitoring of the blue color spectrophotometrically is an indicator for the progress of a reaction utilizing glucose. At certain enzyme assays focusing on the procedure and instrumentation used for such assays. Let us first consider spectrophotometer-based assays. The concentration of a substrate or product can be easily estimated using a spectrophotometer. The absorbance or extinction of a chromogen is measured in a spectrophotometer based on the Lambert-Beer's law, which states that the extinction or absorbance of a chromogen of a monochromatic light is directly proportional to the molar concentration of the chromogen and the path length in centimeter of the light through the chromogen solution. This can be stated as an equation: A, the absorbance, equal to epsilon LC, where A is the absorbance, epsilon is the absorptivity, or L the path length, C is equal to concentration. A spectrophotometer relates the decrease in light intensity of a monochromatic light beam from I0 to I to the absorbance using the equation A is equal to log 10 I0 by I equal to epsilon LC. The spectrophotometer is a sensitive device which can measure concentrations of substrates or products provided they absorb light in the wavelength 200 to 700 nanometers. Micromolar amounts of substrates can easily be measured as the absorptivity or molar extinction coefficient epsilon is usually in the range of 10 into 10 to the power 3 liters per mole centimeter, and absorbance readings typically lie in the range 0.1 to 1 units. The schematic diagram of a spectrophotometer has been indicated. There is a light source. If light is required in the UV region, a deuterium lamp is used. On the other hand, if light in the visible region is required, a regular tungsten lamp is used. The light from the light source goes through a refracting prism and then through a selector, which is in other words a monochromator, and then through the sample. The sample will then allow a certain portion of the light to pass through, which is then picked up by a photoelectric cell and then computed to give the absorbance measurements. A typical spectrophotometer assay is exemplified by the assay for the enzyme acid phosphatase. This is an enzyme present in germinating plant seedlings and in animal cells within organelles called lysosomes. The enzyme is assayed in a fixed time assay in a test tube containing the colorless substrate para-nitrophenyl phosphate and a buffer of pH 5.7. A sample containing enzyme is added. After a period of time, usually 10 to 25 minutes, the assay is terminated using alkali. The role of alkali is twofold. It denatures and therefore halts the enzyme activity, and secondly, the lightly yellow colored product PNP becomes intensely yellow as the resonating phenolate ion created in the alkaline conditions increases the extinction and thus the sensitivity of the assay. In continuous assays such as referred to in a previous section, the reaction mixture is placed in a cuvette, which is a holding vessel for spectrophotometric measurements, and the absorbance is noted with time, say every 30 seconds at a wavelength absorbed by the product. For example, the oxidation of alcohol to acetaldehyde by the enzyme alcohol dehydrogenase can be monitored by following the increase in absorbance at 340 nanometers of the coenzyme product NADH. Let's turn our attention to another type of enzyme assays, which are fluorimeter based. An enzymatic reaction which yields light or fluorescence can be assayed with a fluorimeter or a luminometer. An enzyme called luciferase present in the firefly uses the energy of adenosine triphosphate ATP hydrolysis to oxidize a substrate called luciferin and generate light which can be detected and quantified in a fluorimeter. The reaction has been depicted and you can easily see that the final reaction gives rise to a fluorescent product which gives rise to light which can be modified. The equation indicates ATP plus D-luciferin plus oxygen giving rise to products AMP plus pyrophosphate plus carbon dioxide plus the oxidized form of luciferin and light. This light is typically seen in cold or dark nights wherein you can see the fireflies moving around giving rise to light. This assay is very sensitive as nanomolar quantities of luciferin are required for emission of light easily detected in a fluorimeter or luminometer. In certain assays, the luciferase enzyme is coupled to a gene or protein in a recombinant DNA protocol, and using luciferin in an in situ assay, the location of the gene or protein can be made. A third type of enzyme assays requires the use of a radioactive counter, or these are known as radioactive enzyme assays. Another very sensitive assay for enzymes is based on measurements of product using a radioactive tag. Radioactivity measurements can record up to picomoles of product, making these one of the most sensitive of enzyme assays. The only drawback is that the use of radioisotopes require clearances due to safety issues, and the radiations of some strong radioisotopes may denature or break down other reagents and enzymes. In the assay of the enzyme DNA polymerase, which incorporates deoxynucleotide triphosphates or deoxy-NTPs into DNA polymers using template DNA, radioactive deoxy-TTP, wherein thymidine is labeled in one of the hydrogens with tritium, is added as one of the substrates along with the enzyme DNA polymerase to be assayed, and DNA, which acts as a template, also has to be added. After a given amount of time, the newly formed DNA is precipitated with 10% trichloroacetic acid, and the radioactivity is counted in the precipitate and correlated stoichiometrically with the amount of nucleotides incorporated. The reaction is as template DNA plus deoxy-NTPs, including one of the NTPs which is tritium labeled deoxy-TTP. The enzyme DNA polymerase then strings these together to give rise to DNA, which is now radio labeled. Let me summarize what you have learnt in this module. The measurement of enzyme activity is important to understand the role of enzymes in biology and medicine. Enzyme activity is measured in either international units IU or Ketal units, although the former unit is more used. Different formats of enzyme assays are used, such as fixed time, continuous and coupled assays. The choice is made based on the methods available to estimate either substrate or product in the presence of the other. Spectrophotometers, fluorimeters or luminometers and radioactivity counters are used for the measurement of substrates or products, although the use of radioactivity has been discouraged due to safety concerns.